Okay, I assume we are live now. Let me quickly check from here. Okay. Everything looks good, so we can get started. Welcome everyone for the second lecture of our PNS Genomics course this semester. Today I'll be giving you a slight introduction to the genome analysis. I'll try to show you or motivate you for the importance of genome analysis and show you some important applications to show why we're doing genome analysis. And then we're going to conclude by showing the pipeline of steps, the computational steps in genome analysis, which will then we will continue next week with the initial step, which is the data generation, genomic data generation step. So with that, I believe we can get started. So initially, I want you to recall the slides the slide that I also showed last week, which is we need faster, scalable, and accurate genome analysis, and there are many reasons for that. One reason is it is very important to understand the genetic variations, species, and evolution. It is also important for us to predict the presence of pathogens in an environment, so there can be some organisms that are perhaps harmful for us, and we can analyze and understand the presence of such pathogens in the environment. It is also important for us to, for example, have a surveillance of disease outbreaks, like it happened with COVID-19 very recently. And of course, it is very important for personalized medicine, meaning we can maybe develop drugs that are personalized for you so that we can improve the efficiency of these drugs. So by the way, feel free to ask questions in the chat. I'm taking a look at chat continuously in case if there's any question, and if I don't miss it, I'll try to answer it. So feel free to do it since this is a live lecture. So with that, I'll continue. So then, what is a genome, right? So we're talking about the genome analysis. We're saying that it is important and so on, but it is also important for us to understand what a genome is. The genome, hopefully everyone has it, which sort of is really important, such that it make people look sort of relatively different, right? And also have different phenotypes, not just in terms of maybe their direct look, but also how prone they are to, for example, certain disease, or what sort of basically symptoms that people show given a particular stimulus, let's say. So all of these are mainly determined by the content of the genome. And the content is just basically mainly DNA sequences. Of course, it consists of the other proteins like histones, then chromatins, and nucleosomes, chromatins, and so on. But to simplify and to go in the most lower level, we have these nucleic acid molecules mainly inside your nucleates in every cell. And these molecules are called DNA. And if we're speaking of human, we have 23 pairs. In most cases, we have 23 pairs of these chromosomes. And each pair is coming from one of the parents. We're going to cover these also in the coming slides. So this is basically we can assume for now, in terms of the abstraction level, we have basically these set of DNA sequences in a cell. And if you digitize it, this is what it will look like. So this is a bunch of DNA characters, nucleotides that are essentially ATGCs, right, in the text form. And we want to analyze this. But this is not necessarily as short or as small as this slide shows. It essentially, in a single cell, your genome, especially a human genome, is huge. And to elaborate how huge it is, I'll try to make maybe a weird comparison. So the question is, how large is a genome, right? I'm not sure if you know which building this is, but this building is an Andreasturm, is a tower in Zurich. It's of course multi-floor, multi-level, I guess above more than 18 or 19 floors in this tower. So basically a huge building, at least in terms of the Zurich standards. So if you were to take out every base in your genome for only like one pair of the chromosomes, and if you were to just take out and type every letter in all the chromosomes, starting from the very first chromosome to the very end, up until the very end of your chromosome, the last chromosome, and then you write them on a piece of A4 paper. And then once you reach basically at the end of your chromosome, and then you stack all these papers up together, what you would get basically would be a stack of papers that are as high as this particular tower. And I'm not exaggerating. If you are curious, here I did the math. We assume Andreasturm is around 100 meters. And we can have around like 80 characters per line. We have 4 lines for the A4 page, and each page is around 0.1 millimeter thick. So this is really basically a huge information. So I'm of course saying this just to somehow not necessarily exaggerate, but to illustrate that in terms of the text of information we have, in terms of digital information we have, it's a huge data. And it is only for one pair of your chromosome, and it is only coming from a single cell. So the amount of information actually you're dealing is even larger if you are working either on the multiple copies of the same cell, or working on multiple cells, or even maybe working on even larger genomes, larger than a human genome, etc. But essentially, let's stick with the human genome for now. We know that a genome is important for many reasons, as we covered earlier. And since this is very important, of course, understanding, or let's say decoding the entire genome is also very important. And as the sequencing technologies improved, or as people come up with essentially a working ways, let's say, that can decode the content of your DNA by preserving the order, of course, of the nucleotides that you have in your DNA. People started thinking of decoding the entire human genome. And this effort, although the ways that we can start sequencing the DNA usually started in 1970s, really the effort to decode the entire human genome started around 1990s. And of course, back in the time, this process was extremely slow. You would sort of almost literally have to go base by base, nucleotide by nucleotide, for every position in the genome, and then understand whether this is A, whether this is T, G, and C, and so on. This was really a slow process, and this required also actual human work, let's say, to detect every position of the nucleotide in the genome. So this means that many people were involved, and of course many working hours had to be spent to decode the entire genome, because the entire genome is around 3.2 billion characters, again, only from one pair of the chromosome. And this process started in the 1990s, and then it took around 13 years to complete it. But eventually, we sort of decoded the human genome. As I said, it took around 13 years, but it also cost essentially relatively a lot by spending around $3 billion to decode the first human so this was huge, of course, news, and then this was also on the news and also in the prestigious journals and so on. Actually, the human genome was sort of published or promoted simultaneously both in Nature and Science journals. But that the first decoding of the human genome was not necessarily complete. It consisted of a lot of gaps, a lot of blanks in the genome, because there are also regions in the genome that are challenging to decode, let's say. But as the technologies and the algorithms improved over time, we were able to generate a more complete and almost even fully complete version of the first version of the human genome. This was done a few years ago, around 2021, the decoding of the almost complete human genome. And since this was also was a very impactful work, the time in 2022 selected these 4 individuals that are mainly behind the decoding or the generating the almost fully complete version of the human reference genome. The time announced that these 4 individuals are one of the most influential, 100 most influential people of 2022. So you can see, basically, I'm showing this, you can see the impact, the potential impact of such a decoding, because we want to look at a reference genome, which is a representative of a certain species, and then perhaps identify the variations that compared by looking at this reference genome, comparing your own DNA to that reference genome, and then say whether you have a variation, and then to understand what sort of variation you have, what it means to have that variation, right? So this is one way of comparison. Of course, we're gonna go over with the other approaches where you can sort of reconstruct your own genome and then identify all these variations and so on. As I said, humans have, in most cases, they have 23 pairs of chromosomes. One pair is identified as the sex chromosome, so you either have the XX or XY chromosomes that identifies your gender. And one of the chromosomes, one pair is coming from your one parent, and the other one is coming from the another parent. And if you look more closely, you're going to see this famous double helix structure of DNA where the nucleotides bind to each other. And there is a particular rule for binding. On the one side of this backbone structure, on the one side, you'll have either adenine, thymine, cytosine, and guanine, right? ATCGs, by looking at the initial letters of these nucleotides. But then the rule is, if you have, for example, adenine on the other side, you can only have thymine. Or if you have cytosine on the other side, you can only have guanine, or vice versa. So this is the rule, and I'm assuming that you all know this basic knowledge in biology. And if you look at the DNA under a microscope, this is the structure that you're going to see here. This part is known as the centromere region, and the edges are the telomere regions. And you see essentially the structure of chromosome coming up from the nucleotides, making up basically nucleosomes wrapped around histones, and then chromatins and the chromosomes and so on. This basically makes up a chromosome here. So then we were talking about the human genome. we were talking about the human genome, but let's also talk about the other species, right? For example, here we show a genome or image of a genome from virus. So viral genomes usually have smaller, they are in smaller length, and so in terms of the number of bases, number of nucleotides that they have. Here, this viral genome has around 5.3 kilo or 1,000 bases. If we move a bit further, this is a bacteria known as E. coli, and it's actually a harmful, very harmful version of E. coli strain or E. coli version. Bacteria usually have, of course, genomes of sizes larger than the virus, but relatively, again, in smaller sizes compared to the human genome. In this case, this E. coli has around 5.5 million bases in its genome. Here we have human Homo sapiens. They have 3.2 billion bases, again, from one parent in their genome. So if you're already thinking that, oh, maybe the intelligence somehow, or the level of intelligence or the level of complexity, relative complexity increases as the genome size increases, I would suggest you think again, because now here we have onion, whose genome size is around 16 billion characters. And if you even move further, we're going to see even genomes of larger sizes. So essentially, the plant genomes usually have they are larger than the other species. And the reason for that, in the case of human, we have our genomes are diploid, meaning we have a pair of chromosomes coming from one parent and the other one is coming from another parent. But in the case of the plants, they have genomes of the polyploid structure, where essentially each chromosome has not only have two copies, but have many copies. So that's why essentially their genome size are much larger than the genomes or than diploid genomes. So this is essentially how the genome size differ. But then also, why is DNA useful, or how it becomes useful for the living cells, right? So they are the essential structure of a living cell, because it is important for many cases to essentially, for the regulation, essentially, and also the copying of that cell, and also essentially to function, to provide the correct function of that particular cell, right? And then there are essentially multiple steps where you start from DNA, and then you have this middle step, and then you go up to the generation of proteins, which mainly are the functional part of your of the living cells of the organism. And maybe it's not extremely accurate, but I like to use this analogy where we basically say that maybe the source code, the code that you're writing for your program, is somewhat similar to DNA. And the reason for that, DNA will contain regions that are useful, meaning they are going to be used for the later parts when you're creating the protein. But also there are some regions maybe that they are not as useful, or maybe they are not used for that particular cell. And then this is also a bit, I would say, similar to what you have in source code. You have useful part in your source code, but also you have other parts on your source code that is not useful. And maybe it's just a comment, right? And then when you sort of transcribe your DNA to RNA, meaning when you generate the assembly of your source code, mainly the useful part of your DNA, or the useful part of your source code, will be translated or transcribed, right? So this is then you have your RNA that is coming only from particular regions of the DNA, and then this RNA is mainly used for the synthesis of the protein. And then this is again, I would say, similar to how we execute these assembly codes, right? So they make up the instructions, and then if you are using the CPU, they moved to the CPU, and then the necessary functions are then executed to generate the result that you want to generate. Again, loosely similar to how the proteins are working. So that's why basically I like to use analogy, although of course it's not exactly one-to-one comparison, but maybe to have some understanding, or to create some linking, basically easy linking, if you want to remember how each step looks, and then how we basically move from DNA to protein. So and essentially I said, for example, not all parts of the DNA is useful, or not the entire region of the DNA is used for a particular cell. And then this is basically what makes up, or what differentiates one cell from the other cells, right? So you have your, for example, your liver cell, you have your I don't know, your muscle cells, your skin cells, and so on, right? So they all are somehow different, although their main content is mostly the same, right? The DNA content, because they are mainly coming from your parents, and then initially you start with a single cell, and these single cells grow with the signaling and so on, and then with the signaling and everything, the cells start to differentiate from each other. You make up the brain organs and maybe the other organs and so on, so that, for example, the human body, especially speaking for human, it starts developing. Right? All these basically are happening mainly with signaling hormones, and all the other basically the interactions that is going on when during your development stage. And how this mainly works, without going into more detail, is essentially assume some of your genes that are useful to make a cell maybe to behave like a particular cell, maybe like a skin cell. They are sort of turned off. For example, if you are in the liver cell. And how this turning off works, again, to loosely simplify the process, is they are, for example, wrapped around by certain proteins, so that since they are wrapped, and then since they are not going to be unwrapped very easily, you never express those genes, meaning uh, essentially the molecules cannot go into that region of your DNA and then extract the RNA from that region, which then can make up the protein itself. So they are somehow, let's say, mainly closed for activating that gene. Of course, there are, like, again, other factors, again, signaling and so on. But again, in order for you to perhaps make an easy understanding of how these are working, so you can assume that they are somehow turned off, right? And of course, mutations can occur. Other structural changes can occur. One is, for example, methylation. When methylation happens, for example, a wrapped region in the genome can be unwrapped, so that that gene can be expressed, even though maybe the gene did not mutate itself. Now you can basically express that gene, activate that gene, just because of basically this chemical change, the structure change in the DNA itself. So it's also important to understand the chemical structure, understand these chemical changes, the structural changes in the DNA, as well as the content of the DNA. But of course, they also want to understand the mutations in the DNA, because a mutation can also activate a gene or deactivate a gene, regardless whether it was, let's say, turned off or turned on. And one way of doing this is, for example, we find single nucleotide polymorphisms, or SNPs, which are essentially, just as the name suggests, they are mainly single changes in your DNA at a particular position. And understanding those single changes, small changes, is important because, for example, assume you have these multiple individuals here, and then you sort of try to make an understanding of whether when you have a particular mutation or particular nucleotide at a particular position in your genome, is this somehow related to, for example, the increased blood pressure, right? And then what you can do, you can list all these individuals and then look at the SNPs that they have at a particular position and then say, oh, when I have this, for example, SNP at a particular position, is it related to the blood pressure change? For example, in this case of SNP2, it's not necessarily the case, right? Because regardless whether you have A or G at this position, it can be either low blood pressure or high blood pressure. So maybe that SNP is not really informative for us, at least for that blood pressure. Maybe it is informative for something else. But then you may also identify, oh, actually, there's a particular SNP that is informative, significantly informative for us to tell when you have C at this position, you are likely to have a high blood pressure compared to having, for example, T at the same position. So you can have all these statistical analysis matches and so on, so that you can perhaps link the symptoms, the phenotypes or diseases to these changes. And this is also known as genome-wide association study, GWAS, which means that you have a control group, for example, people with a particular symptom, in this case, people with a heart disease, for example, and people without a heart disease, a control group. And you have also your cases where people show the symptom that you're looking at, in this case, people with heart disease. And then you can, as I said, you can sequence all of their genome and then look at the SNPs, that mutations, the variations that each individual has, and then identify whether the people in this group consistently share particular SNPs with a high frequency compared to the people in the control group, right? And then you can say, okay, if you have the sequence of these SNPs, or if you have only this SNP and so on, you're likely to have heart disease, right? So that's why it is important to understand the content of the genome. And you can, of course, get your genome sequenced, and you can even look at the symptoms potential or the meanings of the variations you have by yourself. Assume you get your genome sequence and then you know which variations you have at what position. And then there are these open databases that you can go and then search to understand what it means to have a mutation, to have a variation at that position, right? And then looking at these variations, you'll get some answers that are not definitive, but will give you some guidance. And we also have much larger variations compared to the single changes or small changes in your genome. The larger variations are called structural variations. These can be large insertions, for example, right? Of up to the theoretical definition of this, like if you have a deletion or insertion or change larger than 50 bases, it's a structural variation. But like this can be as this one suggests. You have maybe 6,000 of characters of deletion in your entire genome. This is a huge deletion, right? And then this can cause, this can be linked to certain, again, symptoms such as autism, maybe obesity, schizophrenia, and underweight, and so on. There can be other types of variations, such as the inversion, the duplication, as I said, deletions and insertions, translocations, where one content actually appears somewhere else, and so on. So understanding all of these is important. And this means that we need to have a clear view of the genome, an accurate analysis of the genome, so that we can understand all these mutations, understand all these changes. But I would say the applications are only limited by our imagination. I'm really, I would say, maybe ambitious or maybe optimistic about this, but I'm mainly positive, right? So I'll tell you many applications that we haven't covered so far, and then you can even, I'll invite you to think even more applications that you can come up with. For example, think about a very fast analysis of genome, right? Maybe even within a second by using limited computation resources. So it doesn't mean that we're going to always have these servers where you can run the huge genome and then get the answers quickly. Maybe we're going to, you want to use your personal computer, you want to use your mobile device, right? And the reason for that, imagine an in-field analysis, a portable analysis, where you have a small sequencer. It's not maybe a huge sequencer. You have a small sequencer that you attach to a drone, and then you let the drone run in the field, right? Or maybe let's say you're hiking, and then you're in the woods, and then you want to maybe understand what type of mushrooms you have in the field. Not the funny ones, but basically the interesting mushrooms. And what you could perhaps do, you would maybe let your drone run, and then maybe with some initial image analysis understands there is a mushroom there, and then goes in and then takes out maybe the extracts the DNA. Of course, there are some challenges of preparing the sample, making it suitable for the sequencer, but there are also efforts in this direction. And then also you sequence and then quickly analyze, oh, okay, this is this type of mushroom. Oh, maybe it's healthy, it's safe to eat, right? So it's almost maybe the most precise way for you to say that this particular thing is safe to eat, right? Not just basically whether it is poisonous or not, but also maybe it will know that whether you're allergic to it. So it's really personalized, let's say, analysis. So it's basically one application that you can think of. But doing also fast analysis is important for urgent cases as well, especially for these critically ill infants. It's like neonates, it's the newborns assume they show a critical symptom, but you don't know what sort of disease that the newborn has, or what sort of treatment that you need to apply. And usually for that, for infants, a type of analysis that we call rapid whole genome analysis is used. Whole genome sequencing is used. Whole genome sequencing means you sequence the entire genome instead of focusing on one region. So you could focus on one region if you knew what you're looking for, or if you want to answer the question whether what you're looking for is there or not, right? And if you know this, you can just focus on one region and then just sequence that region and then do the analysis. But if you don't know what you're looking for, it can be anything maybe, then you need to look at the entire genome, right? And then you need to do it quickly because it can be a life-threatening scenario where you need to answer very, very, very quickly. And nowadays, people are actually doing this type of analysis real time, meaning as the sequencing is happening, the analysis starts as soon as almost the sequencing starts. And the answer can be even given, not just even like within 2 days or 5 days, now actually within hours. Now the goal is actually how you make it even more accurate, right? Because it's a critical answer that you're given, and maybe even cheaper, so that they can be a default coverage, perhaps, for all within all health insurance coverage and so on, so that like every parents do it for their children. Doesn't mean that the child has to show a symptom, right? Maybe it should be default for everyone. So in order to do it, it should be cheap, it should be fast, and so on. So a fast analysis is also important for the rapid surveillance of disease outbreaks, right? Imagine you continuously sequence a particular region, whether there's a harmful pathogen going on. And this has been used for the surveillance of many pathogens or harmful viral genomes before, for example, for Zika virus or COVID, and also for Ebola virus and so on. Genome analysis is also used for testing purposes. A quick example of this is COVID testing. And I want you to remember this painful process that most of us maybe have taken, which is we would have to go through the PCR testing, right? And testing is important because you want to essentially get the spread of the virus under control. And in order to do it, you need to understand which individuals have this virus. And then to do it with PCR, people would take a swab from their nose, and then this swab could, of course, contain your genome, but also the viral genome, right? And if it contains the viral genome, a process known as PCR or polymerase chain reaction would only focus on search for these regions, particular regions in viral DNA, and then would only amplify or extract those regions from the DNA, and then it would make it, let's say, more apparent in the sample. And by doing so, you would then understand, oh, okay, you have this viral genome. But then in order to do it, you will have to wait for some long time so that the amplification and everything happens. But also, it is hard to customize, right? You first need to understand the genome, then you need to know which region in that genome you're targeting, and then you need to basically prepare all these chemical materials so that they attach to that region in the viral genome and then amplify it and so on. So it's also hard to customize. If you want to look at other genome, then you need to do all this process all over again, and then so on. So it also has high latency. People then look at, oh, how can we make it maybe more computational? How can we make it faster? How can we make it even more flexible? And then you can do it with the more recent sequencing devices, where maybe you don't necessarily have to do the PCR testing, or just with some computational approach, you can analyze your genome and then do sort of COVID testing or maybe other type of testing. Of course, the accurate genome analysis is important. It is important for many reasons here. One reason to make, for example, life-critical decisions and improving the quality of life. One very important application that is dear to my heart is genome editing. There are many technologies now where you can do genome editing, but one very famous and known technology is or approach is known as CRISPR-Cas type of editing, where what you can do is you can almost pinpoint to almost any region in the genome. So I'm saying almost, of course, there are restrictions for that, but still almost you can target any region in the genome, and then you can alter the content of that genome. And altering could mean maybe you just delete that region, or randomly change the content with another content so that it sort of turns off the functionality. Or you can even now, there are other approaches, you can even now insert novel bases, novel characters that you want to insert, so that maybe you can create a functionality that you want to create in the genome, meaning maybe turning on the genes so that they start behaving in a different way compared to how they were behaving earlier. And of course, doing all these in a very accurate way is extremely important, because you don't want to alter the genome in an incorrect way, because then you don't know what sort of symptoms that an individual or the cell can develop going into the future. And of course, there are, as you may know, there are ethical discussions about this even today, right? How do you know, even if it is extremely accurate, how do you know in the future and another information will reveal that we didn't know before, and then you didn't make the decision basically based on that information you didn't know, right? So how can you make sure that even in the future there won't be any side effects? And since, of course, this genomic thing has many implications, very impactful, the individuals behind this application that so I'm not saying that figured out the CRISPR functionality, which had been figured out before in 1980s, But what they figured out is actually this is a programmable approach, which means you can sort of program or design the CRISPR-Cas system such that you can target almost any region in the genome. And showing that this is programmable, of course, is extremely important because this means that you can use it for genome editing purposes. And since this has is a huge impact in the field, these individuals, Emmanuel Charpentier on the left and Jennifer Doudna, got the Nobel Prize in 2020 for the paper that they published in, I guess, 2011 or 2012. Basically, you publish a paper and 9 years later, you can even get a Nobel Prize award for that work. So it shows basically how dynamic the field is and how new things can emerge pretty quickly. And of course, we want to analyze the genome in a very large scale. So you may want to analyze a large population of maybe individuals, of organisms, of viral genomes all together, just to check whether there is a particular organism in that environment or how these group of individuals compare to each other or they differ than other population types. So it's large scale analysis is also important. And this is one example where you have essentially a large set of individuals from, for example, Icelanders, and then you analyze them. And then this takes a huge amount of time in terms of the CPR. And think of the energy that was spent. So doing all these in an energy efficient way quickly is also very important. And of course, you usually use cloud solutions. This only doesn't mean that you're spending energy unnecessarily. There are monetary costs implied behind it. So how do you actually do your computation in a much faster way so that these costs are also reduced? And of course, as I said, we may want to do this as even a city-scale microbiome profiling, maybe to understand, for example, here in this case, you're showing Manhattan. You take a sample from maybe a metro station, and then you try to understand whether there's a pathogen that is developing that can be harmful for individuals. And then you want to do this continuously, maybe every day, right? So making analysis faster is really, really important. And also, it may so happen that we may want to do the analysis in challenging environments, maybe even in the outer space, right? How do you essentially take this into account, right? Or how do you develop computational methods that takes this into account? Or even hardware, right? Is hardware more reliable or less reliable in these environments? So all of these are interesting questions, I would say, to ask. And one aspect of course, genome analysis is the privacy. It is a very sensitive data. It is the most sensitive data that you can have, maybe relatively speaking, because it tells almost everything about you, or in a biological way, right? And protecting this is extremely important. So I'm not sure if you know this particular company, 23andMe. It was a very famous and very popular company back in the time. And what people do, people would send their samples. They would take a swab maybe, or take a blood sample, and then they would send it. They would ship it. They would ship their own samples to this company, and then the company would do the analysis on site in their own environment. So you don't know what sort of analysis that they are doing, right? So you don't know what sort of computers that they are using. You don't know the security of their system. But essentially, you're sending your sample to them, and the answer that you get is essentially the insights about your genome, which is actually good, but you don't know whether it is protected, right? And apparently, it was not protected as well. And last year, it was revealed that the data, sensitive data, which is the content of the DNA content of individuals, got leaked. And you don't know whether basically this is in the hands of basically the dangerous individuals that can use it for in an adversary way in the future. And one example would be that I give this example usually, which is imagine that you somehow an insurance company has this data about you, right? And then they already know what sort of disease is that you're prone to. But they don't reveal that perhaps they know that they have that information, right? So you apply for a health insurance and then you get a high estimate. And then you're puzzled. Oh, like how so, right? Why do I have a higher estimate than many individuals? So maybe they know something that you don't know about yourself, right? So all of these, of course, maybe can be resolved by also regulations and so on, but it shows you the potential danger that it can cause to each individual. Or it can be even used for biological attacks, right? They can essentially target individuals knowing that what sort of attacks that a particular population can be prone to. So all of these are essentially very important to protect. And of course, another exciting application is this personalized shopping paradigm. Imagine you go to a grocery store and then you read the barcode of a certain product, and then you want to know whether you're allergic to it or not, or maybe you want to get more insights about this personalized to you. And then you read the barcode and then send it to a company, again, assuming that this is all private. And the company tells you, oh, don't get this product because you're allergic to it. So they can tell you maybe because they know your genome, right? And then they can say, oh, knowing that genome, if you get this product, also knowing the content of that product, if you have this, then you're going to have an allergic reaction or maybe some other things. So you can then imagine how a shopping can also be personalized going in the future. Also, people use DNA, literally DNA itself, for computing purposes. They know that the DNA binds in a certain way, right? So I already showed you the rules of binding, ATGCs, and then they can design the DNA in a way that one end of DNA will bind to the other suffix of another or prefix of another DNA, right? And then they can design it in that way so that the bindings will appear only in a certain way. So this is data encoding. You can encode the data. In this case, this is DNA. You can encode the DNA in a way that they can be used to solve a challenging problem. In this case, there are papers, for example, showing you can encode the DNA to solve the traveling salesman problem in a quick way, right? And this is an NPR problem, but you may have billions of agents, billions of DNA that works in parallel. And what can so happen that if even if one DNA has one type of connection, and then having this connection means that you solve the problem, meaning you know there is a path from somewhere to another node, then this means that you solve the problem, right? So then you can use this DNA to solve all of these maybe hard problems, because it gives you this massive parallelism that the commodity hardware perhaps is struggling to give you. As I said, the applications are limited by our imagination, and I may invite you to even think about even more applications going into the future. And maybe one of the last questions that we want to answer is how and where we can enable fast, accurate, cheap, and scalable genome analysis, knowing that all of these are now important, right? And we know that generating the DNA data, genomic data, is not necessarily a problem. These machines are high throughput machines, meaning they are specialized at generating data at a very, very fast rate. But then how we are trying to analyze this data is via this general purpose machines, right? Your personal computer, or maybe the machines and servers and so on. We're trying to use them for data analysis. And it is slow because these general purpose machines, they do not know the specific requirements of that data, the pattern of that data, the analysis pattern, right? The even pattern of the algorithm that is used to analyze this data. So that's why there can be many problems related to using the general purpose machines, which can make the analysis slow. And there are many reasons why it can be slow. I'll go into that. Even there's now a question on YouTube about this. I guess the question is, why use, for example, an FPGA for genome analysis? What are we currently lacking in the space right now, right? So this is also a related question that I'll try to get into it in a few minutes. So the question is then, right, related to that question is, should we use FPGA or should we use other architecture, or should we use a modern system, maybe a heterogeneous system that consists of many such like different hardware, right? So we could use FPGAs, we could use maybe GPUs, or we could use even the DRAM itself or the memory itself to do the computation, or even the SSD itself to do the computation, and so on. And then we should continuously ask the question, given the type of analysis, given maybe the algorithms that are commonly used to do the analysis, given the type of data, given the requirements of the analysis, what is the right hardware or system for us to use so that we can improve the performance, so that we can reduce the amount of energy that is used, and maybe by keeping accuracy the same as before, even by improving the accuracy, right? So all of these parameters are important for us, and we don't necessarily have to use general purpose machines that rely on CPUs. We can develop our own hardware. And this is even useful for mobile solutions, right? Here, power, essentially, that the devices that you're using are drawing are very important because they have limited memory battery capacities. And to do this, we need to look at basically the entire computer architecture stack from the algorithm down to the devices. And then we need to co-design all of these all together so that we can achieve all of these, optimize all of these parameters that I just told you. And if you're interested in this direction, we have this paper that we provide an overview of the hardware algorithm co-design works in the space of genome analysis, how we co-design algorithm and architecture to accelerate also as well as also reduce the energy and also improve the accuracy in genome analysis. You can take a look at this paper basically, and we are also looking at essentially how we can use new hardware, emerging hardware, to do the analysis for example, newer sequencing technologies. So I'll end the lecture by maybe showing the initial steps of the content of that we're going to cover in the next lecture. How do we start then the genome analysis, right? What are the steps in genome analysis? It usually starts with sample collection. It can be a swab from your nose, it can be your blood tissue. Then we sort of prepare this DNA by sort of essentially maybe attaching so-called adapters on the ends of the DNA so that they can attach or talk to the sequencer that generated genomic data. You can even amplify it. You can even create copies of the DNA if you need to, or maybe you can just focus on a particular region and so on. So this is all library preparation. It's a wet lab process. And we say wet lab because we don't use not necessarily use computational resources here. Then usually DNA is chopped or fragmented into smaller pieces. This is mainly due to either because how sensitive these DNAs are, so it's hard to not fragment them. You need to be extremely careful actually not to fragment those, but also they may need to be fragmented because of the limitations of the technologies that you're using. These fragmented DNA is inputted to the sequencing devices, and then these sequencing devices, then they generate raw data that is typically based on how the sequencing technology is designed. And then this raw data is then usually translated into ATGC so that we can do the analysis. The steps here are mainly we have mainly computational steps, and this is where we are mainly going to be focusing on throughout this course. As I said, the genome analysis pipeline, the common genome analysis pipeline has many steps. It starts from the sequencing data generation up until the downstream analysis, which can generate insights from your genomic data, but after maybe solving certain challenges of the genomic data that we're going to cover in the future lectures. So next week, we're going to cover how we generate the sequencing data. Given a DNA molecule, how do we input this sequencer, and how do the certain sequencers work so that they generate this raw sequencing data, so that we can then continue with the next lectures, how actually we use the raw sequencing data to do the further steps in the analysis. So with that, this is the end of today's lecture. So if you have questions, I'll wait a bit so that you can write it in the chat. If you don't have questions, we're going to end the lecture very shortly. I'm checking the chat now in case there are questions. Yeah, I guess there are no questions. I assume I already answered Raghav's question about what are we currently lacking perhaps in space now. We can use FPGA, of course, if you want to design programmable hardware that perhaps should be changed, how it works later on. And it can, of course, still be more performant than the CPU, right? This is mainly useful for accelerating the algorithm itself in a programmable way. But then what we are lacking is the deep understanding of the needs of the specific algorithm or specific analysis, right? So we need to, for example, reduce the energy. Then this means that is an FPGA really the best solution to it, right? So then we need to show this motivation. So what we need and what sort of hardware can solve this. So you need to create this link, and this link is important. And maybe you could say it could be lacking, right, given an application. But also maybe the reliability of the devices. For example, if you want to use memory-centric devices, maybe they are not as reliable now in terms of the analog computation that they are doing. Essentially, understanding the limitations of the devices is also important, and the limitations of itself of these devices are another challenge. So we also need maybe more reliable, maybe faster and cheaper devices itself. So it's like all basically the entire package itself is like you can just pick an area and then work on this to improve one side of the field. So there are no other questions in the chat. With that, I will end today's lecture, and I hope to see you all next week. Thanks, everyone.